This conference will now be recorded.
Okay. Hi, Vidushi. Good evening, Arsha. So, one thing I wanted to share was that from next week, okay, Anika, fine, you are good. Revise also. Okay. So, from next week, I'll take 3 days a week, and that also already my lectures are from 1 to 1:15 hours, but from next week, I'll increase the duration a little bit because the portion is high and we have to complete it within the limits of the time. Okay, so I hope no one is having any issues with that. Early we finish, more time for revision, better confidence we will have. Fine. Okay, so because when you see this second unit now, this second unit, the topic which we are going to deal now, the cellular compartments, endoplasmic reticulum, if you see mitochondria, cytoskeleton, all these topics, thank you, Anaga. So all these topics are very, very, very broad, okay, and I cannot skip any of the things because these organelles are like the they are the pet questions for the CSIR net. Every year questions are asked from these organelles, and the remaining organelles like lysosomes, peroxisomes, vacuoles, Golgi, all these organelles. Na, see, we are talking today, we will be dealing with the lysosomes. And what is the function of the lysosomes? It is called as the suicidal bag because it clean all the debris and it maintains a healthy environment. Fine. So we will deal with the lysosome. So what I was telling was that these lysosome, peroxisome, plastid, vacuoles, okay, these topics, they have specific questions only which is asked all the time, specific questions. But if you are talking about this cell wall, mitochondria, then endoplasmic reticulum, cytoskeleton, these are the topics which are very big and many of the portions have been asked and much is not touched. So there is always a possibility that those questions can be asked. But if you see these lysosomes and nucleus, peroxisome, plastids, there are not many things to be asked. So it can few important questions are there which is every time which is asked. So my plan is that we will finish this unit within 2 weeks. Okay, 2 weeks maximum. Okay, 2 weeks, so that we can give time to these two parts, because these two parts are very, very big and these two parts are fully molecular, full molecular concepts and everything is full molecular. Okay, so this needs time. Fine. So let's come to the topic. So if we talk about lysosomes, as I have mentioned you, it is called as the suicidal bag. So one question which is asked in your IIT JAM and in your ISC Bangalore exam, which is the suicidal organelle of the body or the cell body? The answer is lysosome. Okay, 1 2-mark question. The answer is lysosome. So what are these lysosomes? These lysosomes are membrane enclosed organelles. Very, very important. They are membrane enclosed because what they are having certain enzymes inside them. And these enzymes, if they come out of this structure, they can be harmful. They can be harmful, but only to those cells which are in a unhealthy condition. Okay, unhealthy condition, or which are creating nuisance to the other cells. Fine. So only those cells will be punished. if this lysosome is present in the normal cell, and by some mechanism, if this cell membrane is disrupted and these enzymes, if they come out, then also nothing will be happening. Why and how, we will see that. Okay, but this is one of the important mechanism which you should know. Okay, this comes for your 4 marks or 2 marks. This question is very, very important, that what happens if this lysosome is broken. Two scenarios are there. One scenario is the cell is dangerous, the cell is unhealthy, and the lysosomes particles are coming out. What will happen in this case and what will happen in this case? Okay, so these are the membrane enclosed organelles, and they contains the enzymes, and the enzymes which they contain have the property to break down each and every biological polymer, be it be proteins, nucleic acid, carbohydrate, lipids, anything. And see, all these things compose the major biomolecules of our cell. Okay, so any of the things, if they are not functioning properly, if they have gone some mutations, if they have gone some other kind of abnormalities, these lysosomes, they have the capacity to digest all these things. It has a very fat stomach. Okay, so it can digest everything. Whatever you give lysosomes to eat, it will eat and it will be very happy because it is having all the enzymes. Fine. The second thing is that now, if you see the lysosomes, they are actually spherical in shape. But when you see the different phases of the lysosome, if suppose you have targeted a lysosome, okay, there is specific receptor by which you can target the lysosome. If you want to see the lysosome in a live environment, you can target lysosome with the help of a fluorescence antibody. Okay, a fluorescence antibody which is targeted to this lysosome. Okay, and you can very well see the full functioning of the lysosome, where it is going, where it is where it is acting as at the endoplasmic reticulum, what function it is playing with the degraded bodies. Everything you will be able to see in this fluorescence microscope with video labs photography. Okay, with time you can see the movement of this lysosome. So when you are seeing that, you can see that this spherical structure is not always spherical, but it is changing its shape, something like this, sometimes like this, sometimes like this. Why? Because at every time it is engulfing certain things, and the shape of this will depend on what it has engulfed. Okay, so the shape is not always the same. So it is a morphologically diverse organelle, a 2 marks question. Which is the morphologically diverse organelle? It is a lysosome. Okay, because they can display variations in the size and shape. Why? Because whatever they have been digesting, they will give them a characteristic shape morphologically. Okay, morphologically. If it has engulfed a large molecule of carbohydrate, so it will be becoming very fat. And suppose it has digested some small proteins, so the size will be not that big, it will be very small. Sometimes it will be shrunken, sometimes it will be very fat. So it is a morphologically diverse. Very important question. Okay, because many times you tend to make mistake with this, because morphologically diverse, if you want to know, you cannot click that it is lysosomes. Understood everyone? No doubt still here. So see, this is your cell, and you can see here, this is the nucleus, the mitochondria, and these are your lysosomes. Okay, these are the lysosomes. And what is present, very, very important, it is a single membrane structure. It is not a double membrane structure. Mark this line, it is a single membrane structure and not a double membrane structure. Okay, and what is present inside this? The enzyme complexes, lipases, nucleases, proteases, all these enzymes are enclosed in this single vesicle bound membrane. Okay, and one more thing you should notice, that from where, a second 2 mark question, a third 2 mark question, okay, from where these enclosings are coming, from where they are budding, from which organelle. Actually, they are a secondary product. Okay, they are a secondary product. Like if you are getting a cell in a nave form, you will be having a nucleus. Suppose a cell is dividing from one cell to two cell, it is undergoing mitosis. So what will happen? It will have a nucleus, a mitochondria, endoplasmic reticulum, and everything it will have. But regarding this vacuoles and this lysosomes, they are made within the cell only. They are not a property which is encoded by your DNA. Okay, and the making the number, the quantity, it will depend upon the surrounding. If you have many viruses, if you have many foreign particles which needs to be degraded, then the number of the lysosomes will be more. And if you have a very healthy environment, then the number of the lysosomes will be less. So they are made from a cell organelle which is called as Golgi apparatus. Can you see the end? The end are budding out in the form of the lysosomes. The endings are budding out. When we are studying the Golgi, we will see that how it is formed. But here you should know that they are made from these Golgi apparatus. Okay, can you see here the budding? And from the terminal end, they are budding into these vesicles. That is why they are small vesicle single membrane bound organelles. Okay, and when these vesicles which are having the enzymes, when they are joining with the phagocytosed material or with the digested material, they will make a bigger structure. Okay, they will make a bigger structure. So when you are targeting this lysosome, you can see each of these phases, when it is coming out, when it is merging, how it is going about here, how it is throwing out all the materials. Everything you can visualize. So next question you have to write in your notebook is that from which cell organelle lysosomes is derived? The answer is Golgi apparatus. Okay, and one more question. What is the membrane property of the lysosome? It is a single membrane bound organelle. Okay, I hope you all are writing the questions. Now, as I was telling you that these lysosomes, they are having, okay Vidushi. So they are having, what they are having? They are having all these enzymes. So what is the function of these enzymes? They will hydrolyze. They will hydrolyze the proteins, the DNA. Very good, Mansa. RNA, polysaccharides, lipids, everything will be degraded. Everything will be degraded. But suppose if there is a mutation in the genes that are encoding, okay, that are encoding these lysosomes, which actually first it is coming into the Golgi, okay, and then whenever the signal is coming, the Golgi is making these lysosomes. How it is making, we will see when we are studying the Golgi apparatus. But suppose from the nucleus, the message is not encoded to the Golgi apparatus. Okay, the genes which are present on the nucleus, they are somehow mutated. What will happen? Or somehow the lysosomes itself have got some problems. There will be a very dangerous situation coming. Okay, suppose if there are some genetic diseases which are happening with the lysosomes, they are called as lysosome storage disease. Very important. Okay, always the disease are asked, always the lysosome storage disease. And why they are called as lysosome storage disease? Because what is the function of this lysosome? They will clear out the stuffs. Whatever the stuffs which are present in the environment, they will clear out. It is something like that. What you call that? The kachra wala, okay, who comes to your home and he takes all the garbage. And if for some days, if he's not coming, what will happen to your surrounding? Everywhere you will be have large heaps of garbage present. Okay, so the lysosome is also a cleaner. It is like a cleaner only. And if it is not able to degrade the things, and because of this, what will happen? All these debris will be stored. That is why it is called as a lysosomal storage disease. Okay, every term has a meaning. And if you decode that meaning now, it is very easy to understand what is the question. So it is called as a lysosomal storage disease. Now, what questions will be asked? The questions will be asked related to the disease. What is the name? The first name you should know is the Gaucher disease. Very important question. It has been asked so many times. Okay, the Gaucher disease. And why it is called as Gaucher disease? Because whenever this type of mutation is happening, it cannot break down this glycoproteins or this glycolipids. So you have to relate it with G is for G. Okay, G is for G because it can be proteins also, it can be lipids also, it can be DNA, RNA, anything. But specifically, this Gaucher is for glycolipids. Understood? It is for glycolipids. It cannot break down the glycolipids. And when these glycolipids are stored in your body, it leads to abnormal adipose adhesions and everything. So it can be very dangerous. Okay, so what is the name? The name is the Gaucher disease. It is a lysosomal storage disease, and what it cannot break, it cannot break the glycolipids. Just this much you have to understand, and this much only you have to remember. Okay, the next disease is the eye cell disease. Okay, the eye cell disease. And why it is called as the eye cell? Just an analogy I'm giving you. Okay, if I'm telling you eye means in our body, it is the organ by which you can see. Okay, so if there is two pathways, and at one road, at one path there is lightings, and at one path it is whole darkened, very darky environment. Which path you will choose? Obviously you will choose the light path. And how you have analyzed that? Because your eyes have sensed that, and your eyes can see that which is the safer pathway. The same is that with this eye cell disease. Now, what is this eye with doing with this lysosome? Suppose when the from this Golgi, the vesicles are coming out. Okay, the vesicles are coming out. So there is a lysosome present. At one phase of the time, there is a lysosome present, but somehow there is a failure of the lysosomal enzymes. Okay, now one thing is that when this vesicles are coming, they will go and target this lysosome only. Only they will go and target the lysosome. They will not go and bind with the endoplasmic reticulum, or they will go inside the nucleus, or they will go inside the mitochondria. No, they will not go to that pathways. Why? Because there is a presence of a receptor which is called as a mannose phosphate receptor. Okay, very important. Very important is this one, mannose 6 phosphate receptor. Let me clear this. Okay, this mannose 6 phosphate receptor. When this receptor is present, it is like a eye, this mannose 6 phosphate, and it will target those vesicles to go to the lysosomes only. That is why I have made an analogy with the eye, and you can remember this very easily when this eye cell disease will come. So eye means seeing, and what is the thing which is helping in seeing is this mannose 6 phosphate. It is a receptor which is present on the vesicles, and this will target it only to the nucleus and not, sorry, to the lysosomes and not to any other place. Okay, so this is leading the pathway. This is, if suppose this mannose 6 receptor is not there, what will happen? A 4-mark question. A 4-mark question. If this mannose 6 phosphate is not present, what is your analysis? What is the conclusion? Tell me. Tell me. Have you understood? Okay, okay. 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 Okay, Asha. I'll tell you. Vidushi, you have given the answer, but suppose the question, but in the question that option is there, fine, and other option is also there that the vesicle will not fuse with the lysosome. The option you have to choose is only with the lysosome, because for every organ there is a target protein. If the target protein is not present, it cannot be directed to that particular organ. Vesicles, if they are not having this receptor now, then also they will not be going to any other stuff, because for mitochondria also there is a specific receptor. If that receptor is present, only then it will be directed to mitochondria. Suppose you all are having a address, right? You all are having a address, Asha, I'm telling. Okay, you all are having the address. Suppose if you have been missed at some place or at other, if you are knowing your address, you can come to your place very easily, right? So what is this eye disease is that eye disease is for something where you have forgotten your address. Okay, what is your address? Your address is I have to go to the lysosome, but you have forgotten. How you have forgotten? Because the vesicles which have to go to the lysosome, which are carrying the enzymes, okay, it has to go to the lysosome. How it is directed to the lysosome? There is a presence of a receptor here, which is called as mannose-6 phosphate. This is a 2 marks question. What is the receptor present on the vesicle which direct it to the lysosome? Okay, so it is mannose-6 phosphate. The name is mannose-6 phosphate. If suppose any organelle or any vesicle or any body, anything which is having this receptor, but unknowingly it is like that, it will go to the lysosome only. Suppose if you have made a body, okay, if you have made a body and you want that it should go to the lysosome only, can you break open the cell and incorporate into the lysosome? No, you have to guide that body to go to the lysosome only. How you will guide that? Just put this receptor, ultimately it will get the signal I have to go here. Understood? Everyone getting? Asha, are you getting the point? So this is the first step for tagging of the lysosomal enzyme. When this tagging is done, only by that it will be directed to this lysosome. So if mannose-6 phosphate is not present, if the vesicle by somehow this mannose-6 phosphate has been cut off, break down, what will happen? This vesicle will stand like that only, and it will be confused. Where should I go? Where should I go? I don't have any address, like that. Fine. So for the address, it should have this receptor. A 2 marks question, and here a 4 marks question analysis can be asked. Am I audible to everyone? Hello? Hannah? Okay. What was the question, Asha? Just a second. Now. Here the message is coming that some technical difficulties are occurring. Just a second. M6P is on vesicle and M6P receptor is on lysosome. Right. Yes. It is not like receptor or like that. M6P is a receptor only which is present on the vesicle, and definitely you can tell this. Okay, mannose-6 phosphate is present on the vesicle and some receptors are present on the lysosome, so they are directed to the lysosomes only. Okay, so this M6P is what is the marker though? So the question will be asked as, what is the marker of the lysosome? If you want to see the lysosome, what is the marker? The marker is mannose-6 phosphate. There is another marker also. This marker is for directing. Okay, this marker is for directing. But when you want to see the lysosome, there is another marker, and we will come to that. So yes, definitely what you told, Nasha, that is one of the possibilities that is happening. Okay. So let's go forward. Everyone is understanding. This is a very small topic, And whatever are the important questions which will be asked, I'm dealing with those points. Okay. Next thing, what is the environment of these lysosomes? Because this lysosome, it is one of the factor which is degrading the things. So definitely it should have a acidic environment. Very, very important. It is having a acidic environment, acidic pH about 5 to 5.5. What is the pH of lysosome? This is the next question which is asked. What is the pH of the lysosome? That is 5 to 5.5, which is in the acidic environment. And the question which I have asked you in the beginning, that suppose in the normal cell, the lysosomes break open, what will happen? And in the degrading cell, if it is open, what will happen? The answer is that when the cells are normal, the cytoplasm pH is in normal condition, that is 7 or 7.2. So when these lysosomes are breaking, this 5 pH will be degraded, and whatever enzymes are present in the lysosomes, they are active only at the 5 pH. If they are coming into any other pH, they will be degraded or they will be denatured. So that is why when in the normal condition these lysosomes are opening, nothing will happen, because the cytoplasm is healthy and it is showing the effect of 7 to 7.2 pH. So the lysosomes enzymes automatically it will be denatured. But what will happen in the abnormal condition? The cytoplasmic pH will change. At that time, the lysosomes, if they are degrading also, they will show their effect. Got the point, everyone? Okay, so that is why this 5 pH now it is giving the double protection. Double protection. If suppose if it is opening also, then also there will be no damage done. And what is the first protection? Definitely it is degrading and denaturing the things which are ultimately dangerous to the cell. So it is having two effect, double protection effect it is having. And how is this pH maintained? Again, a 2 marks question is asked. How is the pH maintained? The pH is maintained with the pump. The pump is called as H plus pump. The pump is called H plus pump, and by now you should remember this H plus pump. It is a energy dependent active pump, and how the H plus is moved, ATP is broken down into ADP and H plus is moved inside through this receptor, through this